The overall goal of the following experiment is to analyze defects in neural development and migration in transgenic mouse models using in utero electroporation of various regions of the embryonic forebrain. This is achieved by injecting a plasmid carrying genes for CRE and a reporter protein like GFP into the ventricle of an embryonic brain and applying an electric current to the head to draw that plasmid into the cell lining of the ventricle. As a second step, the embryos are returned to the dam's abdominal cavity to allow the embryos to continue developing while CRE is expressed and mediates gene excision specifically in the electroporated neurons. Next, embryos are collected and the brains are fixed for analysis in order to determine the effect of CRE-mediated gene excision and subsequent protein knockdown in the electroporated neurons. Results are obtained that show how excision of the gene of interest regulates neural development and migration in a cell-specific manner using immunohistochemistry.
The main advantage of this technique over existing methods, such as tissue-specific transgenic CRE lines, is that electroporation of CRE excises the gene of interest only from a small population of neurons in a region of the brain chosen by the researcher. This greatly simplifies results and facilitates phenotype analysis.
Visual demonstration of this technique is critical because the surgical steps can be very difficult to learn, and the embryos at this stage are very delicate and easy to inadvertently damage.